check. Taking words. Okay, so I think we already talked about the start talk about HDL, right? LDL, finish LDL and HDL, and after watching the movie, I hope you guys have a better understand, the different type of lipoproteins, how they functions, how they regulate the transporter of the lipids, triglyceride, and cholesterol, and how they regulate the the homeostasis of this different type of lipids. And HDL, you already know that it's considered a good cholesterol, or good lipoprotein. That means that the HDL, we wanted to have a higher levels of HDL and have a lower levels of LDL, right? LDL is a bad, the lipoproteins. I mean cholesterol. So the reason, because of these two type of lipoprotein have a different role in terms of the regulation of cholesterol homeostasis. I think I mentioned before, for example, LDL basically is to deposit the cholesterol into the peripheral tissues, and in particular case, in the macrophages in outer wall, which causing the atherosclerosis. And the function of HDL is basically does the so-called reverse cholesterol transport. It means that you remove the extra or excessive cholesterol from peripheral tissue, or particularly from macrophage artery wall, back to the liver, and for synthesis of a bile acid, and then it excreted out of the body. So HDL is a good cholesterol, and we also talk about the core lipids and apoprotein. ApoA1 is unique, right? Is specific for the HDL, and it's synthesized in the liver and intestine. And now we also talk about the reverse cholesterol transporter process. This involved in the alpha HDL and beta HDL kind of interconversion, going cycles. So to finish or to accomplish the removal of the cholesterol from peripheral tissue, to the liver and out of the body. Now let's talk about HDL cycles. So cycles is the interconversion between alpha HDL and beta HDL. So alpha HDL and beta HDL have a very different decomposition of lipids. I added some additional information in this slide, and also next slide I'm going to post it. It's two new slides, like Moodle, right? So for example, beta HDL is considered, some people call it the empty dump truck. That means that the beta HDL only contains these phospholipids, monolear phospholipids, and the ApoA1 apoproteins. There are almost no lipids inside, right? No lipids in the core. So that's why people describe this as empty dump truck, right? Because this the alpha HDL, you can see here the structure contains lots of cholesterol ester inside of the particles, right, in the core. So alpha HDL also considered as a four dump truck, right? So alpha HDL contains the again, similar, the phospholipids, ApoA1, but also ApoA2. And the most important thing is that the lipid core has a lot of cholesterol, which initially loaded from peripheral tissue, right? And also contains some ApoC2. So this is also the new slide, and don't worry too much about it, but I'm going to post it. So just give you sort of a whole picture, how reverse cholesterol transport can be accomplished, and what is the consequences, or what is the function of this reverse transport. So here is the artery wall and macrophages on the artery wall, and this is the beta alpha, the pre-beta HDL and alpha HDL, small and large, right? It also dynamics, the change in the size of the alpha HDL depend on the composition of the lipids and also some proteins. So first of all, beta HDL picks up free cholesterol and phospholipids from the peripheral tissue cells. But here is the macrophages in artery wall, and so the cholesterol are uploaded to the beta HDL, and also this whole process actually happen or take place in the plasma, right? In the plasma. So beta HDL get free cholesterol and phospholipids from peripheral cell macrophages here, and release into the plasma. And in the plasma, there are also the several reactions, occurs, and some changes happen in the plasma. So beta HDL being converted to alpha HDL, and then alpha HDL transport the cholesterol initially from the peripheral tissue and give the cholesterol, particular cholesterol esters, to the liver. So cholesterol esters actually picked up by the liver through this receptor called scavenging receptor, and liver. And in the liver, the cholesterol can be used to synthesize bile acid and secrete into small intestine and the body, right? So this is sort of the whole process. Oh, and then once the cholesterol released and picked up by the liver, then the size is the alpha HDL is converted back to beta HDL because there is no, the lot of free cholesterol esters being released, right? So and then beta HDL then go back to the peripheral tissue and pick up cholesterol again, and then go to the second cycle. So this, as the whole process again, is a process of the reverse cholesterol transport, right? And you can see that the function of this reversal cholesterol transport, or the HDL cycle, is to remove the excess cholesterol from peripheral tissue to the liver and dump it, right, in the form of a bile acid. So I mentioned that the conversion from beta HDL to alpha HDL occurs in the plasma, and several reactions, enzymatic reaction, involve this conversion. And I want you to remember these enzymes, right? It's very important for understanding the process of the HDL cycles. The first enzyme or protein transporter is called ABCA1. ABCA1 is ATP binding cassette transporter A1. ABCA1 is located on the surface of the peripheral tissue cells. right? So ABC1 is a transporter that located on the surface cell membranes of the peripheral tissue. And this ABC1 is very important to transport free cholesterol from peripheral cells and uploaded the cholesterol to the pre-beta HDL. And a second receptor is called SRB1, is scavenger receptor B1. This receptor is located on the liver cells, and it's very important. This is mediating the pickup of the cholesterol esters from alpha HDL in the liver, right? So SRB1 is a receptor that mediates the pickup of cholesterol esters from alpha HDL and to by the liver, right? In the liver, sorry. And LCAT is lecithin cholesterol isotransferase, and this enzyme is present in the plasma. And what this enzyme does is to catalyze the reacidification of free cholesterol to cholesterol esters, right? And there are also the similar enzyme called ACAT, acetyl-CoA cholesterol isotransferase. And this enzyme catalyzes the same reaction, also acidification of free cholesterol to cholesterol esters, but the location is different. This enzyme is localized inside of cells, right? So try to remember these two enzymes catalyze the same reaction, the reacidification of cholesterol, but the location is different. The ACAT is present in the inside of cells, and LCAT is present in the plasma, right? And the other important protein enzyme called CEPT, the cholesterol transfer proteins, and CEPT is present in the plasma, right? And it's very important for the exchange. You remember we mentioned many times, exchange of lipids between HDL and LDL, IDL, right? So again, try to remember this enzyme and proteins and where they're located, what the function, what the function of these proteins.
Can you go back a slide, please?
This one?
The next one? With the whole
one more?
One more back.
Okay.
So I was just looking for the SRB1 was on the liver. I was seeing if there was other.
Oh, you mean this, right?
Yeah.
Yeah, SIB1 is scavenger receptor located in the liver, right, on the cells of liver, and ABC1 should be in the peripheral tissue cell. All right, so now HDL cycles. So first, what happens to the alpha HDL when it becomes beta HDL? Remember, alpha HDL contains a lot of cholesterol esters, right? And should be picked up by the liver. So cholesterol ester inside of alpha HDL is picked up by the liver through the scavenger receptor B1 mechanism. So once the cholesterol esters is picked up by the liver, then alpha DH core volume is reduced, right, because it's loss of cholesterol esters. And the ApoA1, which is on the alpha HDL, is disassociated with alpha DHL, right? So alpha HDL also lose the ApoA1. So ApoA1 is lost, and then internalization of cholesterol from the alpha HDL. And the alpha HDL also contains some triglyceride. So triglyceride is also picked up by the liver together with cholesterol esters, and triglyceride is also hydrolyzed by the hepatic lipase in the liver, right? So in general, the alpha HDL becomes the beta HDL. The size is become smaller because of the loss of cholesterol esters and triglyceride, lipids particularly, and also the loss of the apoprotein, particularly ApoA1. Now, what happened to the beta HDL when it converts to alpha HDL? This is a little bit more complicated, involves several steps in the circulation, in the plasma, right? So first of all, we know that beta HDL is basically is empty, right? Empty from chunk. So basically nothing, there are no lipids in the core. So the concentration of beta HDL should be very high in the peripheral tissue, right? Because its function, right, is to go there and pick up the cholesterol from the peripheral tissue. So beta HDL and the concentration is pretty high in the peripheral tissue, and it accepts or pick up the free cholesterol and phospholipids from peripheral tissues. And then again, I mentioned that ABCA1 is very important, that it localizes in the on the surface of the peripheral tissues, right? The cell membrane, and ABCA1 is the transporter that control or regulates the free cholesterol efflux to pre-beta HDL. That means that ABCA1 is very important, that control the transporter of free cholesterol from peripheral tissue cells to beta HDL. And the pre-beta HDL, once it receives enough cholesterol, free cholesterol and phospholipids, then forming the so-called discodal HDL and re-entering the plasma. Discodal HDL is pretty similar like in that diagram, like a small alpha HDL, right? The small HDL or discodal HDL contains the some free cholesterol and apoproteins, but the size is smaller because in the plasma, the alpha HDL continues to receive cholesterol esters from LDL through the exchange process, right, between LDL and HDL. So what happens to alpha HDL within the plasma? As I mentioned, that the first step that beta HDL receive the free cholesterol and phospholipids from peripheral cells and becomes the small HDL or discodal HDL, and then the small HDL, alpha HDL, continues, receive the lipids, cholesterol esters, and also triglyceride. But some reactions also happen. And remember that the cholesterol first picked up from peripheral tissues is the free cholesterol, right? So free cholesterol, we know that always localized on the surface of the lipoproteins, and this free cholesterol, first of all, need to be re-esterified to cholesterol esters, and then the cholesterol esters is able to internalize to the lipid core of the alpha HDL, right? So this is the important process that characterized by LCAT, this enzyme that is present in the plasma, right? So catalyze the free cholesterol to cholesterol esters, and this process occurs in the alpha HDL on the surface of alpha HDL, and then cholesterol esters be then internalized to the cholesterol, to the core of the alpha HDL, right? And the second thing is that exchange of the cholesterol esters and triglyceride between HDL and LDL, IDL, and VLDL, right? So this exchange and causing the increased cholesterol esters in the alpha HDL, so alpha HDL size become bigger, right? Because it receives more cholesterol esters from LDL, IDL, and even VLDL, right? And this exchanging process is catalyzed by this CPT1 proteins, enzymes, right? So CPT1, the function of CPT1 is to promote or stimulate lipid exchange, right? Cholesterol esters and triglyceride exchange between alpha HDL and LDL, IDL, VLDL, right? So this diagram, I think, gives us a better idea, the whole process, particularly the many steps that occur in the plasma, right? So let's go through the whole process. So the right side, I think, is represented as peripheral tissue cells, right? So you can see the ABCA1 that located on the cell membrane of the peripheral tissue cells, right? And the left side represents the liver, and liver cell surface has the SRB1, right? Scavenging receptor B1, which is important for the pickup of the alpha HDL. And first of all, the beta HDL, which does not contain the lipids, right, and receives the free cholesterol and phospholipids from the peripheral cells, and then it becomes the so-called viscotal HDL or the small HDL, right? Again, the ABCA1 is very important. The transporters that control the movement of free cholesterol from peripheral cells to the beta HDL, right? And once the small alpha HDL is formed, then they release into the plasma. So this whole process occurs in the plasma, right? So first of all, I mentioned that the free cholesterol on the surface of the viscotal HDL need to be esterified to cholesterol ester and then move into the core of the alpha HDL, right? So first the reaction is the reesterification of the free cholesterol to cholesterol esters, and this is the enzyme LCAT. LCAT is an enzyme that present in the plasma and catalyzes reacidification of the free cholesterol, right? And then the second process is also very important, that if the alpha HDL become larger and getting more cholesterol esters from VLDL and LDL, IDL, right? So there's the lipid exchange. So sorry, I think I gave wrong information. So the exchange actually between the HDL, LDL, and it is actually the HDL receives more triglyceride from the LDL, right? VLDL, IDL, and give some cholesterol esters to VLDL, IDL, and LDL. And I'm sorry, I probably need to take my words back. I mean, just try to explain this again. So exchange is that not receive the cholesterol, more cholesterol ester, actually receive triglyceride, right, from the LDL, IDL, and VLDL, right? So initially, alpha HDL, once it's formed from the beta HDL, probably only contains the cholesterol, right? Free cholesterol and acidified cholesterol esters, but does not have much triglyceride. And after it's changed here, then the alpha HDL get more triglyceride from these three proteins and give some cholesterol esters back to this three lipoproteins. So large alpha HDL size is bigger because it gets more triglyceride, right? From these three lipoproteins. So eventually this mature alpha HDL then go to the liver and pick up by the liver. So cholesterol esters be used for bile acid synthesis or steroid hormone synthesis, and the triglyceride being hydrolyzed, right, by hepatic lipase in the liver. Right? Is that clear? Right? Any questions about the cholesterol transport? Okay. Yeah, I think pretty much that's all we need to know about the cholesterol transport, reverse cholesterol transport, right? So for this section that we just talk about, I think what you need to know is first of all, the major components and function of the 4 lipoproteins, including chylomicron, VLDL, LDL, and HDL, right? Again, the chylomicron and the VLDL are the triglyceride-rich lipoproteins, and the main function of these two lipoproteins is to transport the triglyceride, right? And chylomicron basically is transport the triglyceride from the diet, right? Synthesized in the small intestine, and transport the triglyceride from intestine, then absorb it from diet to the adipose tissue and for storage, right? And the lot of triglyceride in the chylomicron is released and picked up by the adipose tissue and stored in the form of triglyceride in the adipose tissue. So chylomicron basically is higher level is increased during the fat condition, right? Because it's needed to transport the triglyceride to the adipose tissue for the storage. And one important enzyme that regulates the chylomicron metabolism is the lipoprotein lipase, remember, right? And lipoprotein lipase is also important for VLDL. And VLDL is synthesized in the liver, and the function is to transport the triglyceride from the liver to the adipose tissue. And also we consider the different physiological condition, right? In the fat condition, the VLDL it basically transports triglyceride from the liver, and this triglyceride initially is converted from carbohydrate, right? In the fat condition, you remember the lipogenesis is activity is high, so and synthesize a lot of fatty acids and triglyceride. So the triglyceride then packed into the VLDL and transported to adipose tissue for storage. And also the part of the triglyceride in the fat condition in the liver that pack into VLDL also comes from Carmichael remnant, right? So initially from the diet. So these two part of the sources of triglycerides is the main contributor to the triglyceride content in the VLDL in the fat condition. And in the fasting conditions, because of the fatty acids level is higher, due to the increase the triglyceride mobilization, right? Fasting, so the liver picks up a lot of fatty acids from circulation, which initially from the hydrolysis of triglyceride in the adipose tissue. So the VLDL and then transported this part of triglyceride basically is to the muscle for utilize, as energy. And also, you guys probably wanted to know that the metabolic fate of lipoprotein, just the whole process, the lipoprotein metabolism. So try to draw the whole process, the pathways, and it probably will help you understand better how different lipoprotein is metabolized. And then we also talk about remember the apoproteins. One of the important apoprotein, ApoC2, right, is on both chylomicron and the VLDL. It's very important that can help the LPL functioning to hydrolyze the triglyceride in chylomicron and VLDL. And also the role of the LDL receptors. So LDL receptor basically is the function of LDL receptor is to help pick up the LDL particles, right, from the circulation into the cells. And the number of LDL receptor on the cell membrane is the key that control the rate of the pickup LDL, then also can affect the LDL levels in the circulation, right? And also we just mentioned that the several factors of protein enzyme, right, that involve the cholesterol, reverse cholesterol transport, HDL cycles. You guys need to know that the enzyme, where they're located, and what is function of these proteins. And also, I think that we talked about this earlier, that the distinct, the different functions of LPL, hepatic lipase, and HSL. Try to make a comparison, enzyme locations and what is the substrate, in which physiologic condition that this enzyme activated and does function, something like this, right? Any questions so far? No? All right, so let's take a 10 minutes break.
Look at the metabolism goes though.
Yeah, right.
I don't know.
Yeah, right. Different ways. I don't know how it works. Sign up, man. Public health.org.
Yeah, so that can this
Okay, so let's get it going. Now let's talk about the regulation of lipoprotein metabolism and some clinical relevance. When you guys know that the lipid metabolism is very close, particularly cholesterol metabolism, right? Very closely associated with the some clinical issues like cardiovascular disease, atherosclerosis. So understand the regulation, how the lipoprotein metabolism regulated is very important concept. So just wanted to go through this composition of a lipoprotein again. See that this gives you a better view, I guess, right? To help you understand, the different lipoproteins, what function, what kind of lipids that they transport and regulate, like collect, the chylomicron, VLDL are the triglyceride riches, right? And lipoproteins, and HDL and LDL again is the cholesterol rich lipoproteins. That means they have a different function. The chylomicron, VLDL basically regulates the triglyceride, right? And HDL and VLDL more related to the cholesterol metabolism. And I put all this five lipoproteins together, in the one slide. Maybe it's too busy, but I just wanted to mention that here the process may not be all occurred in the at the same time, because again, if some process is more regulated during the fat condition, some other fasting condition, right? So just give you here, the purpose of this slide just give you kind of idea. This lipoproteins, and although they have a different function, transport the different type of lipids, but they also interrelated, right? For example, the good example here, the lipid exchange between the different lipoproteins. So they not just completely separate, but they are related, right? So, for example, here the chylomicron, it's synthesized in the small intestine, and the main function is to transport a triglyceride, right? So you once you synthesize the chylomicron in the small intestine and transport the lipids, particularly triglyceride, to the adipose tissue. Adipose tissue is the main destination for the chylomicron. So an LPL again is very important to hydrolyze triglyceride in the chylomicron, pour and release fatty acids, right? And this process again occurs in the fat condition. So fatty acids, once it's picked up by adipose tissue and reacidified back to the triglyceride and stored in the fat tissue. And chylomicron remnant, it can become smaller because a lot of triglyceride pickup is already been transported into adipose tissue. So still some triglyceride that remain in the chylomicron that actually pick up eventually by liver. So liver also gets some triglyceride from chylomicron initially from the diet, and pack it back to the VLDL, right? So VLDL, but in the meantime, because of in the fat condition, we also get a carbohydrate, right? Get glucose. So glucose can be converted to fatty acids through the lipogenesis pathways, and then for the synthesis of triglyceride. So these two sources of triglyceride and packed into the VLDL in the fat condition, and then VLDL and transport the triglyceride from the liver and again to the adipose tissue. So LPL is also important for the hydrolyzed triglyceride in VLDL, right? And VLDL, once it's the the triglyceride is removed from VLDL and it becomes smaller to the remnant, VLDL remnant, which is IDL. So IDL can be further converted to the LDL through the exchange of the lipids. So this between IDL and HDL. So the lipid exchange then means that the triglyceride in the IDL can be removed and transferred to alpha HDL, right? And cholesterol ester that in the alpha HDL can be moved to the IDL. So IDL, basic after lipid exchange, it become LDL. So you can see they have less triglyceride and more cholesterol esters in LDL. So LDL compare IDL has more cholesterol and less triglyceride because it's the exchange of lipids in this process, right? So and remember which protein that involve this lipid exchange process? We just talked about it. Let's go back to a couple of slides. No? Which proteins? Yes, CEPT, right? CEPT, that is very important for this lipid exchange in the plasma. So LDL that gets more cholesterol rich, right? And then it can be picked up by the peripheral tissue and also liver. Right? So liver is the most important tissue that pick up LDL. And then HDL cycle. So beta HDL basically has no colipids and then gets the lipids, free cholesterol and phospholipids from the peripheral tissue, and then after also the reacidification of free cholesterol to cholesterol esters, and also exchange of lipids between this VLDL, IDL, and LDL. Then alpha HDL become larger and mature, then can transport it to the liver. So liver picks up the cholesterol esters and the triglyceride from alpha HDL, right? And to get rid of the excess of the triglyceride from in the peripheral tissue, and alpha DHL and then convert it back to beta HDL once the cholesterol and triglyceride is being picked up by the liver. So this sort of the whole the metabolic pathways of a lipoprotein, different lipoproteins, and the also related, right? Any questions about this slide? No? Okay. All right, now let's talk about regulation. Again, the chylomicron and VLDL basically involve mostly the triglyceride metabolism, right? So it affects more levels of the triglyceride in the body, and IDL, HDL, and VLDL. Sorry, LDL, HDL, and VLDL that the existing kind of triglyceride and cholesterol ester exchange. So this is very important. The lipoprotein that regulates the cholesterol homeostasis or metabolism, but LDL, HDL are the major lipoprotein that regulates cholesterol metabolism. But because the cholesterol, the VLDL and chylomicron is also contains some cholesterol esters, right? And also the exchange of triglyceride and cholesterol ester between this VLDL and LDL, IDL. So the although the VLDL and chylomicron is not a major lipoprotein that regulates the cholesterol homeostasis, but it's also involved and also plays some role, right? Again, this LDL, HDL play a major role, but chylomicron and VLDL also play some role in the regulation of cholesterol homeostasis or levels, right? And this regulation of the lipoprotein metabolism, again, also mentioned, is very closely associated with the dyslipidemia and the metabolic disease like obesity and cardiovascular disease. And in general, the regulatory factor that can influence the lipid metabolism, and including genetic, dietary factors, and environmental factors, right? So I think that genetic and dietary factors are the most important factors that will affect the regulation of lipoprotein and lipid in the body. So dietary regulation of plasma lipids. Dietary fat, particularly composition of fatty acids that are very important, that can affect the LDL cholesterol level and triglyceride levels, right? So again, the dietary fat affect both LDL and the triglyceride levels. And dietary carbohydrate is also be able to affect the triglyceride levels, but not much LDL, right? So again, the dietary fat can affect both LDL and the triglyceride levels, but dietary carbohydrate primarily affect the triglyceride level, but not much effect on the cholesterol levels, right? And composition of fatty acids means whether it's the saturated fatty acids or unsaturated fatty acids, right? We know that saturated fatty acid always is bad fatty acids, have a negative effect, and the polyunsaturated fatty acid like fish oil, and EPA or DHA is considered the good fatty acids, right? You can have a positive effect or beneficial effect on the lipid metabolism. So more specifically, the dietary factors that affect the plasma lipids, the first of all is the percentage of total calorie consumed as a fat. So if the diet contains too much fat as the energy, out of the total energy, that will definitely have a harmful effect. And the proportion of saturated to unsaturated fatty acids. If the diet has more saturated and less unsaturated fatty acids, fat, that will also have a harmful negative effect, right? And then the amount of dietary cholesterol, of course, is also affect the lipid profile. And the major effects, as I mentioned, the circulating triglycerol levels and LDL cholesterol levels, right? Yeah, question.
I have a quick question. How much of an impact does dietary cholesterol play? Haven't they kind of said it doesn't do much?
That's a good question. So I think that the genetic factor probably is more predominant compared to the dietary, right? We know the half, right? Our cholesterol half from diet and half is synthesized from the body. And most important thing is the genetic probably determine how our body is able to regulate or metabolize cholesterol. Right? I think this part is most important. So the genetic, I think, is more important than the diet. So if we have good genes and if we consume a little bit more cholesterol, but be able to metabolize, so it won't cause a problem. So I think the genetic again is a major driver. Right. So average and desirable dietary sources of energy, I think it give us some idea that how we should eat the healthy, right? So average. So let's look at the desirable, but that's right. It's fine. I mean, we can look at average. Average means that in the American populations, you know, how much we consume, the protein and carbohydrate fat. So in the American population, the diet, the 40% of energy is from the fat, and 15% from protein, and 42% energy from carbohydrate, and 3% from alcohol. And if you compare this to the desirable composition of the diet, see how much, how far we need to go to be more healthy, right? So desirable and dietary composition that we want, our diet should have a 30% the energy from 30% of energy from fat, and 55% from the carbohydrate, and 15% from protein. So seems like we consume too much fat, right? A little bit. And then the fat intake, so it's a more kind of fatty acid fat composition in the diet. And the average in American population, we consume the saturated fatty acids, 13% of saturated fatty acids in the diet, and 12% of fat is from the monounsaturated fatty acids, 6% from polyunsaturated fatty acid, right? And also have 2% so trans fat. And if you compare to the desirable, we should reduce the saturated fat, right, from 17 to 10%, and the rest of and maybe also reduce the trans fat from 2 to 1%, right? So again, we consume a little bit too much saturated fatty acids, saturated fat. And carbohydrate, in terms of the composition of carbohydrates, that we have 23% from the starch, 14% from sucrose, and 3% from lactose, and 2% from glucose. And looks like we need to consume more starch, right, from the starch, and then less from the sucrose. Okay, so just give you some idea, the dietary composition that we consumed, and compared to the idea, the composition of diet. Now let's talk about the genetic factors that regulate plasma lipids. That we just mentioned that the genetic factor probably is a major role in the lipid profile and the metabolism. I just give you examples that help our understand how genetic factors will influence the lipid profile or lipid metabolism, right? Here, particularly, we talk about the triglyceride levels, so how genetic factors will affect the triglyceride levels. One example is ApoC2. Do you guys know what ApoC2 does? How ApoC2 functioning in terms of the cholesterol levels and chylomicron levels and VLDL levels. So first of all, tell me what does ApoC2 do in right. How does ApoC2 affect the triglyceride levels? Lowers it? How?
Cofactor to LPL.
Yeah, it's a cofactor of the LPL, right? It's a lipoprotein lipase. And where is the ApoC2 located? In which lipoprotein? We have 4 lipoproteins, right? We just talked about 4, chylomicron, VLDL, okay? So ApoC2 is located on the surface of the chylomicron and the VLDL. Do LDL, HDL have ApoC2? No, right? So try to remember ApoC2 is located on the 2 major chylomicrons and VLDL. And what ApoC2 does is to help LPL and hydrolyze the triglyceride in chylomicrons and the VLDL, right? So if the person does not have enough ApoC2 and their chylomicron or VLDL cannot be normally metabolized, right? So LPL will not have a normal function without enough ApoC2. So how does the ApoC2 deficient affect the triglyceride levels? Increase, right? Yes, increase. So a lipoprotein lipase is not able to hydrolyze triglyceride in chylomicron and VLDL, right? So the triglyceride cannot be break down to fatty acids. So then triglyceride will remain in the circulation, cannot be hydrolyzed without enough ApoC2, right? So triglyceride levels will be increased. And what happened to the chylomicron and VLDL levels? What about the chylomicron and VLDL levels without enough ApoC2? Same thing, right? Because cannot be break down. So ApoC2 deficiency will also cause the increased chylomicron and VLDL levels. Right? So this just gives you the example in how genetic factors will affect the lipid metabolism. Right? So ApoC2 deficient patients usually have a high levels of triglyceride and the high levels of chylomicron and VLDL. We're going to talk about the ApoE deficiency because later, because ApoE is more involved in the cholesterol metabolism, right? So for the triglyceride metabolism, the ApoC2 is a good example of how ApoC2 deficient will affect the triglyceride levels. Okay, now let's talk about the regulation of hepatic VLDL synthesis and secretion. We know that VLDL is also an important lipoprotein that helps transport the triglyceride, right? And the sources of fatty acids that are used for the triglyceride synthesis in the VLDL in the liver. We already talked about it, but here I just wanted to mention again to remind you again that the de novo lipogenesis from carbohydrate, right, is the one of the important sources of the triglyceride in the VLDL, in the fat condition. And I want you to add one more bullet point here, is that the triglyceride from the chylomicron remnant, right, that picked up by the liver is also one of the sources of triglyceride in the VLDL, right? So triglyceride in the chylomicron remnant is another source of the triglyceride in VLDL in the liver. And then the uptake of free fatty acids from the circulation. And this basically happened in the three different conditions. First, the high-fat diet, right? So if we consume a high-fat diet, we have a lot of fatty acids in the circulation that can be picked up by the liver, right? And mobilized. Second is mobilized condition. Second condition is fatty acids can be mobilized from triglyceride storage, which occurs during the starvation. So during the starvation, we also have a lot of fatty acids in the circulation that can also be picked up by the liver, right, and synthesize triglyceride and packed into VLDL. And last is in the diabetic conditions. So diabetic conditions, because of the, remember when we talk about ketogenesis, because of cells are not able to pick up glucose from the circulation and utilize as energy, so body mobilize a lot of fatty acids from adipose tissue and use as an alternative energy fuels, right? So that causing an increase the fatty acids in the circulation. So under the diabetic conditions, the liver also picks up a lot of fatty acids from the circulation and synthesize triglyceride. So these are the also the important sources of the fatty acid, the triglyceride in the VLDL under this fasting condition or the disease condition, right? And increase in synthesis and secretion of VLDL, and these are the conditions that can promote the increase in synthesis of the secretion of VLDL. First is in the fat condition when we consume a high carbohydrate diet, right? And the liver can synthesize a lot of fatty acids from the carbohydrate, and this is the one condition. So once you have a lot of triglycerides synthesized and need to be transported to the adipose tissue for storage, so that this condition can also promote that VLDL synthesis, right? You need more VLDL to transport the triglyceride. And the second condition is the high levels of circulating fatty acids, just like I described in the last slide, three different conditions. Then circulating fatty acids then can also promote the triglyceride synthesis in the liver, and then also that requires more VLDL, right, to transport the fatty triglyceride. So under these conditions, then also promotes, and then ingestion of ethanols and high concentration of insulin and low concentration glucagon these conditions can increase fatty acid synthesis and acidification, triglyceride synthesis, and increase fat oxidation, right? So all these conditions that can promote the VLDL synthesis secretion because of the needs, right? VLDL for the triglyceride transport. So this is just the question that you guys wanted to thinking about in what physiological, which conditions, either physiological or pathological condition that VLDL synthesis and secretion is increased, right? Okay, now let's talk about the fatty liver, the disease condition, fatty liver. So fatty liver means that the liver has an increased triglyceride accumulation, right? So lots of triglyceride accumulate in the liver and causing the fatty liver disease. So triglyceride is a major problems, right? Accumulation is a major problems that for fatty liver. So we know that there is actually a balance between the triglyceride synthesis or formation and the triglyceride export. So in terms of the triglyceride formation, means that the pathway that synthesis of triglyceride from fatty acids, this pathway is contributed to triglyceride formation, right? And triglyceride export means that once the triglyceride synthesized in the liver, that needs VLDL to transport triglyceride out of liver and to other tissues. So if these two pathways as imbalanced, for example, if we have a more triglyceride synthesized in the liver, or we have some problems that export triglyceride, this causing the imbalance, then you have a more triglyceride accumulate in the liver, right? Does that make sense? So this is just the general idea, but it's also some details in each pathways. For example, in the triglyceride formation, not just the synthesize increase, but if we cannot oxidize triglyceride or fatty acids, that also causing the increase the fatty acids accumulation, right? So what are the possible defects that can cause a fatty liver? Given that the balance between triglyceride formation and triglyceride export. So thinking about this questions, and then let's talk about the more information about the fatty liver. So fatty liver can be divided into two categories. One is called non-alcoholic fatty liver. That means that the fatty liver is not caused by alcoholism, and the other type is alcoholic fatty liver. That means the alcoholism is a major causes of the fatty liver disease. So the two main types. For the first type, they increase the levels of plasma fatty acids. For example, the diabetic condition or the chronic intake of a high-fat diet, that we have can raise a fatty acids levels in the circulation, right? So fatty acids can be picked up by the liver and for the synthesis of triglycerides. So this is causing the increased triglyceride synthesis or formation. And the second problem is that the metabolic block of the production of plasma lipoproteins, right? Because lipoprotein is very important for the packaging VLDL, for VLD formation. So in other words, if VLDL cannot be formed, that can also cause the problem, right? The triglyceride cannot be transported out of the liver. So it's another problem that, causing the fatty liver disease. So I guess I'll just give you kind of brief, concept of a fatty liver disease. I think we probably should stop there, and we have a informal writing today, right?